Thank you. So, hi everyone, Shalom. My name is Shira. I'm a postdoc at the lab of productie, the Broad Institute, and I really appreciate the opportunity from Tal and Raz to speak about our recent project on SARS-CoV-2 antigen presentation. So I will start with a brief introduction on this, one of what I think is the most elegant systems in our body, which allow constant presentation of the cell content to the adaptive immune system. So while the virus is outside the cells, it can be seen easily by antibodies. However, when the virus enters the cell and starts replicating, the adaptive immune cells, or specifically T cells, cannot see the virus directly inside the cell. And the way that they engage the cells are infected with the virus is by this antigen presentation pathway. So briefly, proteins are constantly degraded by the proteasome into shorter peptides that are translocated through the Tap transporter to the ER, where they can be uploaded on MHC class 1 or HLA-I in human. Then this complex is presented on the cell membrane. And as long as this peptide is coming from endogenous protein, there will be no T cell response. However, in case of viral infections, some of the proteins in the cell will come from the virus, meaning that the peptides can result from a viral proteome, and this will be identified as a foreign peptides and allocate the T cell response that will result in eliminating the infected cell. And this process has been thoroughly investigated, but mostly focusing on the canonical open reading frame of what we know is translated from viral genomes. And we start to appreciate in the last few years that there is much more substrates for this presentation, including non-canonical open reading frames, such as alternative reading frames, upstream open reading frame, and short open reading frame in non-coding RNAs. And it was shown in a handful of cases that these short open reading frame can indeed induce peptides that are successfully presented on HLA and can invoke T cell response. And here, for example, you can see the T cell response in patients infected with CMV that can recognize these short peptides coming from what we used to call a non-coding RNA in CMV genome. So with that, my research in the past few years were focusing on two main questions, trying to understand better this question and try to understand the complete repertoire of antigens available for presentation and for us to understand the complete signature of viruses as seen by the immune system. First, what is translated from viral genomes, which we don't know for many, many viruses. And second, which viral peptides are presented on cytotoxic T cells. And while working on this project and developing the experimental systems, this happened. And this is the reason that we're having this lovely seminar today on Zoom, and we're all aware of the ongoing COVID-19 pandemic. And these questions that were highly relevant to understand general principle in viruses became very relevant and concrete for specifically SARS-CoV-2. So we might can understand better the interaction between SARS-CoV-2 and the immune system. And importantly, a recent study from the Stern-Genosaro lab at the Weizmann Institute identified that in addition to the 12 annotated open reading frames in SARS-CoV-2, there are 23 non-canonical open reading frames, some of which exceed the expression of some of the structural annotated proteins in SARS-CoV-2. And while we don't know their function, it seems that we should take them into account when we think about the subset for antigen presentation. So with that, we started a project that we're aiming to profile the HLA presentation in cells infected with SARS-CoV-2 with three key questions in mind. First, which viral peptides are presented on HLA in infected cell? And specifically, whether the 23 recently discovered open reading frame can contribute to this repertoire of presented antigens. I would just note that current vaccines and immune monitoring approaches focus solely on the canonical open reading frame. So if there are any peptides coming from this region, we are completely ignoring them for now. Another question is whether SARS-CoV-2 can interfere with, in general, with the antigen presentation pathway. As we know, some other viruses do, such as viruses from the herpes virus family and HIV. And unfortunately, I don't have time to get into the finding of this aspect, so I will just give a small spoiler and say that our findings suggest that the SARS-CoV-2 can interfere with this process, mostly with by interfering with enzymes that regulate upstream processes to the proteasome degradation may influence the reduction of the pool of peptides available for presentation. So in order to ask these questions, we performed the following experiments. We infected cell in BSL-3 facility with SARS-CoV-2 in a high multiplicity of infection. We infected both A549 cells and HEC23 cells, and then we allow enough time for proteins to be translated and processed and presented on HLA. And then after 12, 18, and 24 hours post-infection, we performed 3 functional assays. First, we pulled down the HLA complex and determined the sequence of the presented peptide by mass spectrometry. We also did all proteome analysis using mass spectrometry to determine the host response. And finally, we did RNA sequencing to determine viral abundancy cell. And the key point for the pipeline of searching these data sets in the mass spectrometry data is that in addition to the human proteome and the annotated proteome of SARS-CoV-2, we also appended the non-canonical open reading frame, allowing us to identify any non-canonical peptides from this region. So this is just a brief summary of this experiment. So on the x-axis, you can see the position across SARS-CoV-2 genome. Over here, you can see the 12 annotated open reading frame of SARS-CoV-2, and they are colored here according to their expression levels. In the track below, you can see the position of the non-canonical open reading frame that were identified by ribosome profiling in the stern-Gannon-Star loop. And the next 4 tracks represent the HLA peptides that we identify either in HEC293 cells or in A549 cells. And there are many interesting insights coming from these results, but I chose to highlight one striking observation that we had, which is 30% of the detected HLA peptides that we observe are actually derived from non-canonical open reading frame. And more specifically, they are derived from out-of-frame open reading frame in the coding region of the spike and the nuclear protein. So you can see these 2 regions here magnified, and you can appreciate the position of this non-canonical open reading frame in both these regions and the position of the peptides that we found. And I will just note that these open reading frame are out of frame, meaning that they are encoded from the same region, but they're encoded in a different frame, meaning a different amino acids. And it has consequences when we think about potential approaches for vaccine design. So we know that the spike, for example, is one of the key targets for current vaccines. And we do not know the sequence of the RNA vaccines currently available, but we know that a very common approach when you try to increase the expression is to use human codon optimization. And human codon optimization is a very powerful tool because you can increase the translation of your gene of interest while maintaining the sequence of the open reading frame and maintaining the sequence of the amino acids. But this is true only if you look on the main open reading frame. When using human codon optimization and changing the codons, you probably alter the sequence of any alternative reading frame if existing from this region. And this may lead to attenuation or even destroying these peptides that we detected. So in order to appreciate the effect of human codon optimization in the spikes on the production of these peptides, we look on a publicly available sequence that went through human codon optimization from the Krogan library. And it's not surprising to see that it has the exact same amino acid sequence in the region and code the spike. And this is what we are doing when we optimize codons. However, if you now look in the +1 frame, which is the frame that encodes this alternative non-canonical open reading frame, you can see that the sequence is quite different. And in fact, if you zoom in into the regions in which we identify these peptides, you can see that their sequence is quite different. And this suggests that while trying to optimize the expression to increase antibody response, unknowingly, we might have destroyed the sequence of important peptides, potentially compromising the T cell response. And this is true for SARS-CoV-2, but in general, we know that many viruses use overlapping open reading frame. And we know that they do that in order to increase the coding capacity from what is relatively a very small genome. So we suggest that maybe this is a more general principle, maybe we need to take it into account when we think about vaccine design. And it might be that if we try to optimize a sequence to increase the expression of a specific gene of interest, let's say in this case, the spike, in order to increase the antibody response, we might unknowingly compromise the production of peptides from alternative open reading frame that can be really fruitful substrates for T cell recognition. So for regions in viral genomes that encode these overlapping open reading frame, we might have an inherent trade-off between optimizing the antibody response and optimizing the T cell response. And in order to take this into account to many viruses, kind of like circling back to the beginning of the presentation, we need to know more about the translation and presentation in viral genomes. So with that, I would like to end. And I want first to thank Raz and Tal for this really great opportunity and putting together this amazing symposium. From my lab, Productie Betty, which is an amazing and inspiring mentor, Charlie Rice, who served as a really thoughtful advisor throughout the projects, including this one. Musin Said did all the BSL-3 work, and from the mass spec platform at Abroad, Susan and Jen, Sissy from New York Cohen's lab. And this is the funding of that project and my funding for the postdoc fellowships. And thank you all for bearing with us. I know it's past 8 p.m. in Israel, and I'll be happy to take any questions.
Thank you very much, Shira, for this really great talk. Do we have any questions from the panelists?
Shira, that was a great talk. I was wondering if you do phylogenetics of the virus, are there any signs that the virus is gradually optimizing its codon usage to be more similar to human, because it's a relatively new human virus?
Yeah, so it's recently spilled from bats, so it's kind of hard to do this analysis. I will say that, you know, we kind of like wonder why we even need to do human codon optimization, why the virus is not already optimized if it's, you know, circulating in human. And I will say that when you talk about optimizing things for expression for vaccine, it might be different than optimizing fitness. So the virus might not want to have too much spike, and we know that too much spike expressed on cells can cause cell syncytia in tissue culture. And you need to also take into account the stoichiometric ratio that the virus needs for all the structural proteins. So from the virus point of view, you know, the virus needs a specific amount of spike, not necessarily the highest amount that you can reach to with human codon optimization. When we try to think about the vaccine, we really want to induce more from this specific protein. It's outside the context of the native virus and the stoichiometric ratios that you need between different proteins. So this is a kind of like a different question.
Okay, thank you. We have a question from the crowd. Ranit Kedmi, I'm going to let you speak if you want to ask your question.
Do you hear me?
Yes.
Okay, great. Hi, Shira, great talk. I was wondering, in the context of the symptoms of this disease, I was wondering if you was thinking to check if there's some kind of similarity to human peptide that can explain maybe the symptoms, some kind of mimicry between the open reading frames you see to self.
Yeah, so we started to look at it. We're kind of like wondering in general whether we can find, you know, when you look viral peptides, how similar they can be to endogenous peptides, which might suggest, you know, in the context of the vaccine, it may even suggest some side effects of autoimmune disease, right? So
yeah.
So we are now at the steps of trying to get more clinical samples. And the first question in mind is, we do see this presentation on HLA, and we see this in vitro. And the next challenging question is whether it even invoked T cell response in patients. So we have very preliminary results from a few COVID-19 patients that we see response against these non-canonical peptides. We haven't got into the details of characterizing the response according to severity of disease. We will need more samples and try to understand this process better. One of the key points might be similarity to human genome. I will say that when we look for the data sets in the mass spectrometry, we also search against the entire human proteome. So it didn't come up as something that is similar to human proteome, but as we don't know about the entire repertoire of viral open reading frame, we also don't know about the entire repertoire of human proteome as well. And this is what I guess the scientific community trying to advance using ribosome profiling and other approaches.